Good day, everyone. I'm Shalline Thangadurai. Today, I would like to present my research findings about the transcriptome analysis of Ctpsyn induced testicular tumorigenesis in Drosophila melanogaster. Ctpsyn aids in de novo synthesis of the nucleotide CTP, and it compartmentalizes into filaments called cytophilia. These cytophilia are conserved across prokaryotes and eukaryotes. Interestingly, cytophidium formation negatively regulates Drosophila CTP synthase activity, which is opposite to the human CTP synthase. Higher number of CTP synthase cytophilia is related to various types of human cancers. As you can see here, there are 5 phases of cytophidium assembly, and in the Drosophila egg chambers, only one isoform, which is isoform C, is observed to form cytophilia. As mentioned before, increased CTP synthase activity is a feature of many types of human cancers. When CTP synthase was knocked down in Drosophila cancer models, tumor formation was observed to be reduced. Furthermore, filamentous structures were detectable only in cancerous hepatocytes. In a recent finding, overexpression of CTP synthase isoform C alone was observed to be sufficient to induce testicular bulging in the flies. What could be the pathways or genes that leads to this testicular overgrowth in flies? As you can see in the image here, the testis is bulged with the transit amplification region filled with cytophilia in a CTP synthase overexpressed flies. The objectives of this research are: perform transcriptome profiling on CTP synthase overexpressed line and identify and verify potential genes or pathways responsible in the tumor-like formation. Here I show the transcriptome analysis pipeline that I have used. Mapping using RNA star and differential gene expression analysis using HR were performed with Galaxy Bioinformatics platform. This data shows the alignment scores for the mapped reads. Above 85% of the reads were uniquely mapped with the Drosophila genome for all the samples. This figure shows the volcano plot of differentially expressed genes. Those red dots on the right shows upregulated genes. Meanwhile, those on the left shows downregulated genes. Here is the heat map showing the clustering of the top 100 differentially expressed genes compared to the control and the CTP synthase overexpressed flies. Gene ontology for biological processes shows that most of the differentially expressed genes are involved in immune response related processes. As you can see here, antibacterial human humoral responses, defense response to gram-positive bacterium, humoral immune response, response to bacterium, innate immune response, defense response to bacterium. All of this falls under this immune response related processes. This figure shows the gene ontology for cellular components and followed by gene ontology for molecular functions. This figure shows the KEGG pathways that are regulated. As you can see here, TOL and IMD, apoptotic and Fanconi anemia pathways are interesting to be studied further as they are involved in immune responses in Drosophila melanogaster. These are the immune response related genes that were differentially expressed. To validate these genes, qPCR was performed. All the apoptotic and autophagy related genes were significantly downregulated. MEI41, decay, TRAF4, and ATG9 are genes that are involved in apoptotic and autophagy related pathways. Evasion of immune response is one of the hallmarks of cancer. In a previous study, the impaired human Ctpsyn synthase 1 has been identified to cause immune deficiency. Hence, studying the interaction between Ctpsyn synthase and immune response could assist in a better understanding of cell proliferation, and tumor formation. Autophagy and apoptosis, both these are tumor suppressor pathways. Interestingly, genes involved in these pathways, MEI41, decay, TRAF4, and ATG, are significantly downregulated. This could lead the tumor cells to escape the surveillance and thrive in the tumorous microenvironment. However, further studies need to be done to validate this. Thank you very much for listening. If you have any question, you may ask. Thank you.